Hello, everyone. Thank you so much for being here. I'm so happy to see all these smiling faces here today. I'm here today to talk about my dissertation that I've written. For the sake of today, I've retitled it to Everything You've Ever Wanted to Know About Tillers But Were Too Afraid to Ask, because that's what we're going to be talking about today, of course, right? So before I start, I want to dedicate my dissertation, this presentation today, to my family and all of my friends and all of the wonderful people and families who have made it possible for me to be here today. So, okay, we are going to look through a snapshot of my dissertation. We're going to be talking about 3 of my main chapters. And we're going to do an introduction. I'll do a quick summary of the field studies because we use the same field studies for all of my chapters. We'll go through 3 chapters that I've selected for my dissertation and then some general conclusions at the end. The first chapter of the introduction I want to go through with you first is crop plasticity. And as we know, this is the ability of a genotype to express different phenotypes based on the environment that it's growing in. This is an innate characteristic of plants because plants were faced with the option to either adapt to the environment that they found themselves in or die over time. And as we went through the domestication process, these plasticity mechanisms have been suppressed or removed in some cases. And in our modern genetics, the modern crop species that we have, the extent and the expression of these plasticity mechanisms vary based on the crop you're looking at. Broadly, we can categorize these plasticity mechanisms into source and sink. And source, what I'm talking about there, is energy accrual. So examples of that would be the number of leaves, the size of the leaves, the morphology of the plant, the architecture of the canopy, all of those types of things. The sink, we're talking about where that energy is going to, what it's being used for. And examples of that would be our yield components and our root structure, the examples of that. So tillering, of course, is the plasticity mechanism that I'm going to be talking to you about today. And tillering is essentially a branching response in grass crops. Branches grow from the crown region at the base of the plant. And it increases both source and sink potential because we're increasing the number of leaves. We're increasing stem tissue, which is a transient sink, right? It kind of acts as both. And the grain set is increased. The potential for that is increased because we have more shoots that can form on. Tillering is not viewed the same in all grass crops, as we know. So if we talk about wheat or grain sorghum, which are very common in Kansas, we might find management recommendations based on the ability of these plants to tiller. We might manage 4 tillers. That could be a component of our strategy. You will never, ever hear someone make a statement like that when we're talking about corn. Corn puts tillers on, but they don't always put grain on. Some people might argue that they rarely put grain on, depending on the situation you're working with. But tillering in corn, they historically have been called suckers. And something that I found really interesting is looking at the literature on this that was published in the early 1900s. These scientific articles were calling them corn suckers instead of corn tillers, like in the published literature. And I thought that was just really interesting. But these tillers, the ability to put on these tillers has been masked over time through genetic selection and also farmer preferences with how they're managing their fields. We'll talk about plant density in just a second, but that's a great example of that. We can still see tillers in modern hybrids if those plants are exposed to certain environmental conditions. And because tillers are kind of an oddball thing in corn, it's generally an unstudied response. And the yield reports and conclusions that are out there are really mixed and sparse and don't agree very well. So there's a lot of opportunity to improve the knowledge that we have on tillering in corn. Talking specifically about plant density in corn, the genetic improvement that we've seen to push yields higher in corn is tied very strongly to plant density. And so you'll see here, we have plant density on the x-axis, grain yield on the y, and the lower curve here with these circles, these are hybrids from the 80s and 90s. And the box across the top is our 20 teens varieties. You'll notice that our plateau on this curve moves further and further to the right over time. So we're increasing these plants' need for higher plant populations to reach the yields that we want. So very dependent on that. But here in Kansas, we have some unique challenges because we are not the I states, right? We can't manage our corn so intensively. And there's a decent amount of corn that's produced in this area, but as you'll see, the yields are lower. The yield environment is very different. We have to manage it differently, and plant population or plant density is one of the challenges that we have with this. So optimum plant density is a word that we like to throw around. It's difficult to figure out what that is until the season is over. It's kind of a magic number that no one really knows what that is, right? And it varies seasonally. So you'll note here, we have some ranges in suggested plant densities from a publication from K-State. It ranges a lot based on the region of the state you're in, but then also within a region like the Southwest, for example, we see that there's a pretty big range in this recommendation as well. So if we could reduce the dependence of these corn plants on the plant density in environments like this, that could be a useful trait for us for managing this corn. So with that in mind, the general objectives that we're going to go through today are to determine the impact of tiller development on corn yields, the drivers and predictability of that tiller development, and then the effect of tiller expression on reproductive efficiency and carbon economy. So as a brief overview of our field studies, we establish our plots in a three-way factorial treatment structure with 3 plant densities, 2 genotypes, and 2 tiller treatments. So tillers were either left intact, so we didn't touch them throughout the season, or we removed them at stage V10, and we'll talk about that a little bit more in a second. But we used a split plot arrangement and a randomized complete block design with 3 to 4 replications depending on the site year that we were working in. That's a lot to absorb. So we're going to look at a plot aerially to see what that looks like. So this black line delineates the extent of our study. This is in Garden City, Kansas. And we have 3 blocks going across this field. In each block, we have strips of plant density. So the 25, 42, and 60,000 plants per hectare. Layered on top of that, we have our genotype, so P0657 and P0805, both from Corteva AgriScience, commonly planted in this region and prone to tillering, which for a tillering study is probably pretty important when you're picking a genotype, right? And then layered on top of that, we had tiller removal treatments. So we either left the tillers intact or removed them. And that was the layout of our plots. The tiller removal was done at stage V10, which is the 10th leaf in corn, and that was done by hand. And the reason we picked V10 was a compromise between avoiding ear development at V12 and on. We didn't want to mess with that as much as possible. And then avoiding regrowth of those tillers, because at V10, if tillers were present, the tassels on those tillers will have initiated. So breaking the growing point off of those tillers means that they weren't going to grow back. Because as you'll see in our map in a minute, going to all of our sites to make sure we didn't have more tillers wasn't going to be an option. So the data we collected was separate for tillers and main shoots. Everything was done separately. We took phenology and stands at multiple time points throughout the season. And then biomass samplings were done at 5 stages, V5, V10, V16, R3, and R6. Those biomass samples, the stem tissue that we collected was saved and then analyzed for water-soluble carbohydrates in our lab. And then the grain yield was hand harvested to make sure we could separate where that grain was coming from. And our environment characterization was done through soil sampling and then online weather information. So our site years were located across the state. The points that you see here are the locations, and the number on each point is the number of years we had at each of those sites. So total we had 17. You'll see that they were spread across the state pretty well. Of those sites, we did intensive biomass samplings at 6 of them. We picked the sites in the corners of the state to try to capture as much environment variation as we could with that. The Far western and southern sites, these purple ones here were limited subsurface irrigation. They were established on the Corteva yield test sites, and they have this limited irrigation capability basically to make sure that they have something to harvest at the end of the season and they're not throwing away a bunch of money on their yield test investments. So we had those on those fields, but this was 2019 to 2021, so across 3 years. Our environments, the normal seasonal rainfall ranged from 330 to 550 millimeters, and then our seasonal temperature from 19 to 22.5 degrees Celsius. Our yields ranged, the mean yield at each site ranged from 6 to 12 tons, which is less than 100 to over 200, if that conversion to English units is less comfortable for you. The soil characterization was accomplished with 15 and 60 centimeter cores at V2, and then our soil physical properties were downloaded from the web soil survey based on the coordinates of each site. A couple metrics before we jump into the good stuff. So the climate data was taken from Climate Engine for the period of 1991 to 2021 so that we could calculate the 30-year normals for all of the variables we were looking at in these sites. Our water supply included precipitation and irrigation if it was applied, and the full season irrigation or the full season water supply, we considered 1 month pre-plant as a way that we could have a measure of how much moisture was in the soil at the time we planted, because we didn't take actual soil moisture measurements. The growing degree days, again, were added from planting with the 10 and 30 degree limits. And then one that may be less familiar for some of us, the photothermal quotient is essentially a measure of solar radiation over a given time period. And in our case, that would be megajoules per square meter per degree day. The basics of this is if you have a higher photothermal quotient, you're going to have more energy available to that plant during a given time period, and the opposite if you have a lower photothermal quotient. So the second chapter of my dissertation, we're going to talk about the effect of tillers on corn yield. This is published in Crop Science. We also put out an extension bulletin on this, but essentially this was our 2019 and 2020 sites. So this is 10 site years total that are included in this first chapter. Our hypothesis was that corn tiller expression would alter the yields of the corn plants that they were appearing on. And our objectives, which I'm framing as questions to help us follow through these, were: will tiller presence impact yields? Will the number of tillers change that relationship? And then why are tillers more productive in some site years than others? So we're going to go through those one at a time. For this first question, we conducted our statistical analysis in R. We fit linear mix effects model with treatment factors, site year, and interactions as fixed, and our design factors as random. Our response variable, of course, with our grain yields. And after we fit that model, we conducted a type 3 analysis of variance with the 0.05 significance level. And what we found is plant density and genotype were both significant at the 0.05 level. Essentially, yields increased with greater plant density, which based on the graph we saw earlier, we would expect. The genotype, it was significant, but the effect size that we saw was not large enough to really be meaningful. It was very negligible, the numbers and the difference between those yields. The 0.001 significance level, we found that site year was significant. Of course, environment has a lot to do with how those plants respond. So again, the 6 to 12 megagrams per hectare range. Site year by plant density was significant with only one site not impacted by increasing plant density. So that means that you could add more plants, but you weren't getting more yield because that environment was restrictive enough that we saw that. The site year by tiller status interaction, 2/3 of our sites had no change, whether tillers were intact or removed. The other third did, and those are the ones we're going to talk about. So in this plot, you'll see the two highlighted panels are the site years where we had a significant impact of tillers. The TI on the x-axis indicates plots with tillers intact, and the TR, those with tillers removed. The grain yields are on our Y, and you'll notice that in both of these site years, Garden City and Goodland, we had a significant increase in yield when tillers were present in both of these sites. So will tiller presence impact yields? Yes, it can. but tillers did not reduce yields in any of the evaluated scenarios that we tested. Plant density and environment, as expected, were important to determine the yield potential of these locations. And then greater genetic diversity should be included in the future to see if including more hybrids could change some of these relationships that we saw. So we said that tillers present positively or neutrally impacted yields, but does that change based on the number of tillers that you have appearing? So again, linear mixed effects models, instead of our treatment factors, we use our shoot count. So this would be the total number of tillers, the total number of main shoots, the actual numbers that we had there. Site year interactions with our design factors, again, is random. And we found that all of those combinations were significant. What that looks like, so on the x-axis here, we have our observed plant density in plants per hectare. On the y, the tiller density, 1,000 tillers per hectare. The black points that you see on this plot indicate our observations. The black dashed line is the maximum number of tillers that we would expect to observe, because as we increased our plant density, the number of tillers went down. The gradient shows us the yield, the predicted yield. And so what we can see in this example in Keats in 2020, there is a strong gradient as we go from left to right, indicating that a lot of the yields were being pushed by the number of main shoots that we had. As you go up and down, you can see that there's not much of a gradient in this direction, so tillers weren't having much of an impact on the yields that we were seeing in this site year. If we look at these other two examples, Colby B was the site where we saw no impact of plant density on yield. You'll see that regardless of tiller or plant density, there wasn't much of a change that we saw in those predicted yields. But Goodland, which is a little more interesting, we have a gradient along the x-axis, and we also have a gradient along the y-axis. And what that tells us is that there is some potential of tillers to match yields at a lower plant density with a higher tiller density to match a higher plant density with no tillers. You can see that. So will the number of tillers change that relationship? No, we did not see a negative impact of tillers within reasonable tiller densities. The tiller effectiveness did vary by environment. We did see that. So more tillers didn't always equate to more yield, which can raise some efficiency concerns that we'll address later. But tillers could compensate to some degree for fewer plants. The maximum yield was always with the highest plant density. So we did still see that tillers could help, but they weren't replacing adding more plants. Why are tillers more productive in some site years than others? So why did we see that change across environments? We conducted a principal component analysis, and our clusters were based on the responsiveness of each site to tiller density. We took the tiller density coefficient from the model that we just talked about and clustered our sites based on that. The parameters that we looked at were water supply, photothermal quotient, our minimum and maximum temperature, a range of soil variables that we had collected, and then the periods that we were targeting for this were pre-plant to V4, which is before tillers would be appearing on those corn plants, V4 to V7, which is during tiller appearance, those tillers are actively being put on, and then V7 to V14, which is when those tillers would be developing. And what we found is in the first PCA dimension, we had 36% of the variation that was captured there, and that was explained by soil variables. The second dimension, we had a quarter of the variation captured, and that was our photothermal quotient and our minimum temperature. So essentially, what this means is that yields were most responsive to tiller expression when we had higher silt and clay percentages, higher organic matter, and a higher seasonal photothermal quotient value. And yields were most responsive when we had a lower minimum temperature after those tillers had appeared. So why are tillers more productive in some site years than others? We found that the photothermal quotient is a novel link to tiller responsiveness in corn. It's been linked to tiller productivity in other crops, but not in corn previously. And these additions to the carbon reserves in the main stock, because the photothermal quotient is a measure of the energy that that plant has available, So understanding how the carbon reserves change will be explored in a chapter that we'll address in just a bit. But tillers have been identified as potentially able to translocate the carbohydrates from themselves to the main stock for grain filling. So we'll go into that later. So again, we had our hypothesis that tiller expression would alter yields. We looked into that hypothesis with these three main questions. And the takeaways that we have are that tiller presence never reduced yield in any of the environments or scenarios that we evaluated in this study. In sites with good soil quality and high fertile quotient, corn tillers had the ability to compensate for lost plant density. And an adjustment was always needed in our plant density to maximize yield. Tillers could help, but they're not a replacement for good plant density management. So the next question that I'm sure you're asking is, how can we accurately predict the number of tillers we're going to see in a field? You show me that the number of tillers can impact the yield response that we see. So can we predict that? Well, you're in luck because we're going to talk about it. So predicting corn tiller densities, this is currently under review in the European Journal of Agronomy. Essentially, our hypothesis was that those tiller densities could be predicted within 25% of the plant density that we were targeting. And our objectives, again, framed as questions for us to go through. Which variables are key to predict tillers accurately? And then how useful are these predictions in new seasons? Before we get into those questions, I need to talk just a little bit about the field data, because the way that we treated the data for this chapter, it was an observational study. So we basically took tiller counts in the plots that didn't have tillers removed. So we kind of ignored our treatment structure when we were taking this data. We took tiller counts at various points throughout the season, but I just wanted to make a note of that before we get into this too far. So which variables are key to predict tillers accurately? We took the data that we had collected across 3 seasons. We're talking about 2019 to 2021 now. And we had 15 model candidates with 16 variables that included environment, management, genotype, and then various combinations of different data that fell into these categories. We made our selections of these candidate groups based on our prior knowledge, so what was published in the literature, the observations that we had made in the field ourselves, and then how easily producers could manipulate some of these variables that fell into these categories, because we wanted our research to be as usable and meaningful as possible. So we tried to consider that when we were grouping these candidates. So our model, we use a generalized additive model, and our effects were fit as smoothing functions with thin plate splines in the MGCV-R package. We added a degree of biological constraints through our selection of a distribution. So we use the binomial distribution, which essentially gives us a 0 to 1 probability of success. And in our case, we define success as a maximized tiller density. And you may be asking, what does that mean? We had to come up with that. So our theoretical maximum tiller density that we decided on was 300,000 tillers per hectare. And that was based on a maximum plant density in modern corn plant density studies of 100,000 plants per hectare, and then a maximum reported number of tillers at a field scale of 3 per plant, which, as you can see, we did in our field trials. For some context, though, the current study, the maximum that we saw was just shy of 153,000. So a little ways away from the 300,000 maximum, but that was good for us because that meant that we weren't constraining our model by selecting a bad maximum. So essentially what this model is doing, it's giving us a probability between 0 and 1. We multiply that by our maximum, which is 300,000, and that would give us a number between 0 and 300,000 as the number of tillers that we would expect to see in a field that we're working with. We used out-of-sample validation to evaluate this model. So 80% of the data was provided as a training set that was randomly selected, and 20% was a testing set only. So the model did not see that data when we were training it. We only used it to test how well it would perform. The predictive accuracy was quantified with the mean absolute error, which is essentially the absolute value of the predicted minus the observed value. And then our target error was 25% of the plant density, as you'll recall from the hypothesis, which meant that based on our average plant density of 42,000 plants per hectare, we were wanting an error. We wanted our error to be less than 10,500 tillers per hectare. So how did we do? The best model candidate that we had was an E by M, so environment by management model with these variables included in it. It was interaction between plant density and each of the following. So plant density by growing degree days, plant density by vapor pressure deficit, etc. This model candidate gave us a mean absolute error just shy of 8,000 tillers per hectare, which if you've ever seen corn tillers in a field and how sporadic that can be sometimes, I was very surprised and happy with this error that we were able to achieve with these models, which is less than what we were targeting. So what does the background of this look like? So we have smooth effects. So essentially here we have growing degree days, but we have our range of observations that we had for growing degree days. You'll notice that our y-axis goes from 0 to 1. And the reason for that, if you'll recall, is we have a probability between 0 and 1 that we're multiplying by about 300,000 maximum. So growing degree days had a very clear non-limiting threshold. So above 200 degree days, there was not a limitation by growing degree days on the number of tillers that we could see in this field. If we look at something like the vapor pressure deficit, we have a minimum, but then as we get into our higher plant densities, we also have a maximum where these plants are more likely to be stressed based on this higher vapor pressure deficit, and we can see tiller abortion in these higher plant densities, potentially due to vapor pressure deficit. We did have some soil, the nutrient variables that were significant in this model, but our relationships were a little messy because we did not have dosed nutrient treatments at each of our sites. We only had the observation, so the whole field nutritional information. So more specific studies would be needed to identify clear thresholds in this regard, which is a limitation that we had with this data. The last question, how useful are these predictions in new seasons? So we took the GAM that I just showed you, the best candidate from that first part of the analysis, and we kept that structure, but what we wanted to do is see how well it would perform if we showed it a new season that it had not been trained with. So essentially what we did for each of our years, so say 2019, we took our 2019 data and put it through a model that had been trained with our 2020 and 2021 data to see how well it would predict. We did that for all 3 of our years. And that changed our train and test split a bit. So in 2019, we did keep an 80-20, but in 2020 and 2021, that shifted. So why were we doing this? The reason is because we wanted to see how well this model would perform in a scenario where it was given data that it was not trained with. So a new season, as we all know, every season is different. And so just to see how sensitive our model was to something like that. Again, mean absolute error was used to quantify accuracy, but then we wanted to know what variable is causing the most problem. So which one is the least calibrated, the one that's giving most of the error. And so for that, we selectively zeroed one coefficient at a time to see if removing which variable would improve our error. So which one is causing the most problem. Now, of course, as we would expect, the accuracy of these models is very dependent on the data that was passed to them. Because if a model is given data that it has not seen before, it's not going to know what to do, right? So if we look here, we're going to compare our 2020 and 2021 sites with the error. There's quite a big difference, as you can see. So the black dashed line here at the base is our 10,500 target for our error. The 2021 locations you'll see danced around that line pretty well. But if we look at 2020, there's obviously something that's not right, right? Something's not working the way it's supposed to. So the most improperly weighted when we did our out of season tests were variables associated with tiller abortion. So either the model was saying you're aborting too many tillers late in the season or you're not aborting enough, or the fertility variables, which was our nitrate and phosphorus, because again, we didn't have those dose studies, so those thresholds were a little fuzzy. In 2020, for example, which is a site we just looked at that had the crazy high error, those sites had both the lowest and the highest fertility in our data set. So that would be a reason that it was really out there. So again, our hypothesis, corn tiller densities are predictable within 25% of the target plant density. We explored these two questions. The take-home messages from this chapter are that tiller densities can be reliably predicted with environment and management variables that we identified. Weather thresholds in our study appeared more clear, but that's because we don't have good dosed data on our fertility variables. And the importance of a representative training set is key to this. We saw that it can change your results a lot based on what you're feeding your model. So based on this information that I presented, in season or diagnostic type applications will probably be the most realistic. So a farmer wants to know why they have more tillers one season than another. These could be some variables that you could look at to try to answer that question. So the next question that you're probably asking is that we saw that not every tiller produces grain. We've talked about that. And the question becomes, how is that impacting the efficiency of these plants? Because if you have a tiller that's not putting on grain, what good is that doing? So we're going to talk about that. This paper is currently under review in Field Crops. And our hypothesis going into this chapter was that corn tillers would improve the ability of the plant to capture carbon and the efficiency of that plant based on the fact that you're improving the sink potential, but that would hinge on the ability of the tiller to successfully produce grain. So the questions that we're going to go through: do corn tillers reduce whole plant reproductive efficiency? And do tillers increase carbon reserves or impact the remobilization of those reserves during grain fill? So the first question, this analysis is much easier to follow. So we fit linear models in R. We had three main things that we were looking at with the harvest index. So the first one here, the main harvest index related to the main above ground biomass production as a reference value to see how things compared to that. Tiller harvest index to the tiller biomass, and then the total harvest index, which would be main shoots and tillers compared to the tiller biomass that was developed. So on the x-axis, we have our above ground biomass, which is for main and tillers. And then we have our harvest index on the y. The lavender points are our values for the main shoot only. And our dark purple points are for the tillers only. So the main shoot, so just considering what was growing on the main stem, the harvest index was fairly stable across increasing biomass. The tiller harvest index, however, as tillers increased in their biomass, the harvest index skyrocketed. And that makes sense because if you think about the way tillers develop in corn, if they remain vegetative, they appear, they stop growing, and they don't put grain on, right? But if they're able to reach reproductive phases, they're able to continue growing and developing, they're putting on more and more biomass, they're more likely to put grain on, which increases our harvest index in that regard. So just to explain those relationships a little bit. But the last one, we have our tiller biomass, above ground biomass, on the x-axis and our full harvest index on the y. And we can see that there was no relationship between the amount of biomass that our tillers were putting on and the harvest index of the plants that we were looking at. So regardless of how much biomass or how large those tillers were, we weren't seeing a reduction in the full harvest index of those plants just because of the tiller biomass. So do corn tillers reduce whole plant reproductive efficiency? In our case, they did not. Corn tillers had no impact on the full harvest index that we saw in any of our sites. Tiller and main shoot harvest index both had similar potential. We saw values up to 0.6, which is very high. That has been reported previously for tillered phenotypes, though, so that's not totally outrageous. The harvest index was not reduced by the fact that we had tillers there or by how large or how many tillers were growing on that plant. The second question, do tillers increase carbon reserves or impact remobilization during grain filling? And just as a quick review to make sure we're all on the same page, water-soluble carbohydrates are stored in the stem in corn. The corn stem is a transient sink. So the sugars are stored in the stem and then a portion of them can be remobilized during grain filling. And in this way, the carbohydrates in the stem were used, in our case, as an indication of the energy needs and the source sink balance in that plant where things are moving and what's happening. So the seasonal water-soluble carbohydrates were fit as a flexible, smooth defect of growing degradase with the smooth spline function with cubic splines in R. And we fit these lines separately for tillered and non-tillered plants, as well as separately by plant density. So we're going to look at these figures. We have our three panels for our three plant densities. The x-axis is our cumulative growing degradase, so this is our season duration. The y-axis is our water-soluble carbohydrates found in the stem in grams per plant. The gray area that we see here is our cumulative water-soluble carbohydrates as the water-soluble carbohydrate dynamics as we go through the season for plants that had tillers removed. So this would be main shoots only. If we look at our other line, our dark blue dashed line, we can see that this is the dynamics of those water-soluble carbohydrates over the season when we had tillers present. So this is the main shoot and tillers, all the carbon that you had in that plant in the stem. And there's a few things I want you to notice. The first is that if we draw a line across the peak of the water-soluble carbohydrates in the stem when we had tillers removed, it does not matter what plant density you have. There is a strict cap on the amount of water-soluble carbohydrates that were accumulated in the stems of these plants. If you add tillers, which the biomass tissue was a lot of stems in these tillers, so we added a lot more potential to store carbon. We actually, in our lowest plant density, were able to escape that cap and almost double the amount of carbohydrates that we had in our stem reserves in these tillered corn plants. So do tillers increase carbon reserves? Yes, we found that tillers increase those reserves with that added stem tissue in our lowest plant densities. The tillers could overcome those strict caps that we saw on the water-soluble carbohydrates per plant. And then this buffering of the water-soluble carbohydrates is significant agronomically because if we have less strain on the reserves we have in those stems, it increases our standability. So we're less prone to lodging in that way. And then if we encounter stress later in the season, it can help to stabilize our kernel weights because we have more reserves to pull from. The last part of that question, we fit linear models again, considering the main stem, water-soluble carbohydrates, and then the main grain biomass on these plants. We fit that for tillered phenotypes and non-tillered phenotypes. And then within the tillered phenotypes, we were looking at barren and grain-bearing tillers. And that's a lot to absorb. So to recap, we are comparing source, so tillered versus non-tillered, and we're comparing the sink of that tiller, so grain-bearing tillers versus barren tillers. Now, if we look at this plot, we can see we're looking at our non-tillered phenotypes first. So we have our observations and then our line here. We see that there is a slight trend upwards, and our highest yielding or the main plants with the largest sink had a larger proportion or a larger amount of water-soluble carbohydrates remaining in the stem at harvest, which sounds a little bit backwards maybe. But if we think about this, this was most likely tied to the amount of water-soluble carbohydrates that these plants had early in the season. They had a greater supply to begin with in this case, but it shows us that these plants are potentially sink limited, right? They have extra carbon. So if we look at our tillard phenotypes, we see in the dashed line and open circles are plants that had barren tillers, so tillers did not have grain on them. The solid line and our solid points are our plants with fertile tillers. And what you'll notice is there was no significant trend comparing our main stem reserves in this case. And actually, our plants with barren tillers had a higher average yield sink size than our fertile tillers did. And we're going to talk about that in just a second. But essentially, to answer our question, do tillers impact remobilization during green fill? We found that yes, tillard phenotypes can stabilize the water-soluble carbohydrate remobilization because we're softening perhaps that sink limitation that we were seeing. Both barren and grain-bearing tillers behave similarly. They both had those similar lines. We also have data for R3, which I did not show today, but the relationships remain similar. The tillard phenotypes also appear to maybe be more productive when tillards are barren rather than when they have ears on them. And there's an important caveat with that because tillers are notorious for putting on tassel ears. Not every ear that a tiller puts on is going to have grain on it. So if we were to take that apart just a little bit more, we might find that that could be a contributor to that relationship that we're seeing. So our hypothesis, corn tillers improve plant carbon capture and efficiency, but this hinges on successful tiller grain production. We went through our 2 questions to explore that. And our take-home messages here were that tillers had no effect on reproductive efficiency. In this case, we were looking at the harvest index as a measure of that. Tillers improved the plant level ability to store carbon. We saw an escape from that cap on our stem water-soluble carbohydrates. The barrenness of the tillers, so whether or not the tiller had grain, didn't impact the harvest index or carbon economy relationships. We actually found that they could be more productive potentially if those tillers are barren. And as with everything that I presented today, all of these relationships could change if we had a more restrictive scenario with less water, with different conditions that we didn't explore here. But what I've shown helps to explain the stabilized yield results that we saw in the first chapter. It connects very well. So what's next? We're going to go through some conclusions quickly here. So the main take-home messages, if you were enjoying your food and missed something I said and would like 3 things to take home with you today, I have them here for you. So tillers did not reduce corn yields in any of our trials. This includes our 2021 sites, which I did not present because our first chapter was over just 2019 to 2020, but 2021 held the same. The environmental factors that we've talked about, the impact, both the number of tillers that we see and how well those tillers do for us, how much grain they're able to put on, how productive they are. Tillers can also benefit the carbon reserves of that main plant and can stabilize the source sink balance that we talked about as well. 2 things that I would like to leave with you as far as remaining questions. There's so many more questions. We could talk for days about tillering if you want. But the reproductive development of those corn tillers, so how do those tillers develop? Because as I mentioned, corn tillers are notorious for putting on these nasty things called tassel ears. They develop, this is actually taken from inside a tiller as these are developing. You can see we've got silks growing, we have tassels developing, it's kind of a mess. But understanding when we're going to get a tassel ear on a tiller versus this normal development that leads to ears that we can harvest, right? All the ears you see here, like 5 ears in this picture are on tillers. So they can put ears on, but when and why is something that we're still not sure about. And the other part would be, how can we take this information and put it in a way that's usable for farmers? So these 2 pictures here were sent to me by farmers on Twitter. This first picture, he had plants that were hailed out really bad early in the season. They ended up putting a lot of tillers on because the main shoots died. And then in this picture, there were some rows with some pretty severe planter skips, and we had a lot of tillers that were coming out in this situation as well. So replants or different unexpected scenarios that happen after we plant that we're not able to mediate, how can we provide some kind of value or management recommendations or different things based on what we talked about. With that, I would like to thank you all very much for coming in, for all of your attention, and I would welcome any questions, comments, or snide remarks that anyone has about anything that I've shared today. Thank you.